Thank you very much, Oskar, for agreeing to give a Build a Cell seminar, and you can get started.
Thank you very much. So welcome, everyone. My name is Oskar Staufer. I'm not anymore at the University of Oxford or the Max Planck Institute in Heidelberg, as the invitation said. I'm now an independent researcher at the Leibniz Institute for New Materials in Saarbrücken, Germany, in the Pharma Research Alliance with the Helmholtz Institute for Pharmaceutical Research and the Center for Biophysics at Saarland University. And today I would like to talk to you about the bottom-up assembly of viral replication cycles. So essentially, this will be a first, not build a cell, but actually build a virus talk in this seminar series. And I've been thinking about how to motivate this for the audience. Yeah, that is mostly centered around looking at how cells divide, at the fundamentals of metabolism in cells, membrane biophysics, and so on. And I did something that I think many of us have been doing in the last days and weeks. I got lazy and I started to use ChatGPT and see what actually ChatGPT would say when it was asked to motivate such a talk, and when it was asked why we should at all follow research that is aimed towards the bottom-up assembly of viral replication cycles. So the answer I got is pretty much well aligned with what I think. So I actually just copy-pasted this, and I think it gives a pretty well summary of the motivation behind this line of research, which is essentially the idea that when we build something like a synthetic virus that is able to replicate inside host cells from well-defined molecular building blocks, we'll be able to understand in more quantitative detail how virus replication infection progresses and evolves, but also how a host cell is actually impacted by this. The methods that we would use to create a particle such as that depicted on the left side here, which is a primitive artificial virus infected a mammalian cell. The methods that we would use for this are pretty much the same methods that we use for the bottom-up assembly of artificial cells, only that we deal with much smaller vesicles. And these are also methods that would not only concentrate on non-living building blocks, but also in the interface of living and non-living building blocks. And what we would gain from this is essentially a high degree of control over the structural functional composition of these viruses, which should actually enable to get us more detailed understanding for processes that are relevant for biomedical applications, gene therapy, and so on. So why at all is it actually a good idea to target viral replication cycles with bottom-up assembly methodologies? Well, if we look at the virus infection cycle, as here depicted for the SARS-CoV-2 virus, we can essentially see that this is a modular process in itself. We can identify very distinct processes happen in the extracellular and intracellular environment that will allow for the propagation of the virus in a host. And I just want to summarize these individual modules for you, which essentially start by a module that's usually found on the outside of a cell that allows for the identification of binding sites, specific binding sites on the surface of host cells. That's essentially a module that would bring specificity to the infection process. This is followed by a second module that would initiate the uptake of the virus particle into the cellular space, and which is essentially well connected with the third module that allows for the release of genetic information from a virus into the cytoplasm. And these first three modules are very distinct from the subsequent three modules, which are essentially more based on the processes intrinsic to the host cell. The fourth module would essentially initiate the engagement of the transcription translation machinery of the host cell in order to read the genetic information and reprogram the biochemistry and the cellular biology of the host cell in module 5 in order to induce the assembly of new viral capsids, which is then in the last step followed by the controlled exocytosis or release of newly formed virus particles into the extracellular space to initiate this viral replication cycle again. So as you can see, the first three modules could in principle be quite easily engineered from outside of the cell, but once inside of the cell, module 4, 5, and 6 would heavily rely on genetic engineering approaches and other top-down approaches. And I think this is one of the interesting aspects of this line of research, which is when we try to build artificial viral replication cycles, we not only need to follow bottom-up strategies, but we need to work hand in hand with top-down conventional genetic engineering approaches that would allow us to initiate reprogramming of the cell. And I will specify these modules later on. I will give you one specific example of how this process can be initiated, especially concerning module 1 and 2. Okay, so as I said, this is also not only basically our opportunity to bring bottom-up and top-down approaches together, but it's also a prime example of how we can use bottom-up synthetic biology to look deeper into mechanisms relevant for biomedicine. And as such, I want to give you one prime example how in this line of research of bottom-up assembly of viral replication and the use of bottom-up synthetic biology, we can actually get biomedically relevant insights. And the story I'm about to tell you starts in the very early month of the pandemic, essentially when we were collaborating with the labs of Imre Berger and Christiane Schafitzel at the Max Planck Center for Minimal Biology in Bristol, which were back then, and I'm talking about the summer of 2020, working on solving the structure of one of the most important proteins of the SARS-CoV-2 virus particle, which is the spike protein. They resolved the structure of the spike protein, not as the first to ever do so, but actually in doing so, they found something that all the other structures have overlooked until then, which is that in the spike protein, there is a fatty acid bound, specifically linoleic acid, an unsaturated fatty acid. And the special thing about this discovery was that when this fatty acid was bound into the spike protein, this binding seemed to lock or stabilize a closed structure of the spike protein. Now, many of you are now almost experts, I would say, in SARS-CoV-2. At least that's what the last 3 years of pandemic have brought to us. But I still want to introduce some of the basic aspects of the spike protein to you in order to show you how we use this to build or to establish synthetic viral replication cycles and how we use such a primitive viral particle to understand the function of this fatty acid binding pocket. So if we look at the spike protein of SARS-CoV-2, we can essentially appreciate that it's a trimer made out of 3 single amino acid chains. And this complex molecular machine can have different configurations. Essentially, we can discriminate between an up and a down configuration. The up configuration, essentially that configuration that will allow the spike protein to identify and bind to the host cell receptor, the AC2 receptor. And in that up configuration is essentially that form that will allow for the binding. However, if the spike protein is down, the protein is not able to bind to the AC2 anymore. So it's a non-infectious state of the spike protein. And what we found and what we reported back in the autumn of 2020 in this paper is that the fatty acid binding will essentially stabilize a closed structure, and was completely unknown at that point why the virus should at all have a fatty acid binding pocket that will essentially limit its infectious potential. Although back then we also didn't have any real methods and means at hand to understand this. So the questions that we not only got during the revision process, but essentially after making this discovery public was what the actual function of that fatty acid binding pocket is. There are not too many fatty acid binding pockets in viruses around. It was not clear which kind of fatty acids could bind and why and if this fatty acid binding pocket would be relevant for an infection. So why did we need to develop and apply a bottom-up approach for this? Well, essentially, conventional top-down approaches did not allow us to dig deeper into these questions and understand the function of the fatty acid binding pocket. Of course, it is possible to produce in cell cultures virus particles that would be loaded intrinsically with fatty acids, but unfortunately, it's not possible to remove these fatty acids after the viruses have been produced, simply because the methods that would remove hydrophobic components such as fatty acid would also destroy the viral envelope of that particle. At the same time, we can simply not produce and work with fatty acid-free cultures simply because fatty acids are abundant molecules present in each cell and in each culture medium as well. So this was also not the route to go. And this was also the moment we decided to follow a minimal biology, a bottom-up biology approach, and start to construct artificial viruses from scratch. Essentially, we started again from cell cultures, but not producing whole viral particles, instead just recombinant spike protein loaded with fatty acids, from which we could then produce an apo spike protein, essentially a protein from which all fatty acids have been removed by standard preparative chemistry approaches, so essentially hydrophobic columns. And from this then produce something that we call minimal viruses, probably more specifically minimal virions. So virions are the particle state of a virus that is essentially built from a lipid envelope onto which the recombinant spike protein is immobilized. So the story that I'm about to present you focuses on the first two modules of the viral particle assembly and of the initiation of this viral replication cycle, and will later on focus more on the other modules from 3 to 6 in the discussion section. So, of course, producing these vesicles is, for us as a synthetic biology, bottom-up synthetic biology community, an easy thing to do, right? We deal with lipid membranes almost daily. We have very good methods to produce very well-defined lipid membranes. And as such, it was easy for us to adapt our methods from our synthetic cell assembly to start and produce minimal variants of SARS-CoV-2. Essentially, we produced SUVs with a defined lipid composition that resembled the lipid composition of living, in quotation marks, living viruses. So a complex mixture of approximately 9 lipids, and we produced them with a comparable size to those found of the natural SARS-CoV-2 virion. And onto that vesicle, we immobilized the recombinant SARS-CoV-2 spike protein. So when these vesicles were incubated with human ACE2 expressing cells, we could very nicely see that they actually attached to the cell surface, and that this attachment was most likely initiated by ACE2 binding, at least when we were comparing minimal virions that had the spike protein on top and such virions that lacked any protein on the outside, so essentially naked lipid vesicles. We could see that these vesicles with the spike protein on top start to accumulate in the cell cultures by binding to the surface. So we use this admittedly simple system to start and look deeper into the function of the fatty acid binding pocket. We had some knowns from the structure, some things that we actually knew. For instance, we knew that the fatty acid binding pocket that you can see here essentially featured a kink, and that kink in the pocket was very much pointing towards a concept where only unsaturated fatty acids could actually bind into that pocket. So fatty acids that feature an unsaturated carbon-carbon bond that would induce kind of a kink in the fatty acid itself, and only those would actually fit in the fatty acid binding pocket. So we selected 4 fatty acids with different saturation stages, and we measured in our retention analysis, which is basically a measure of how well these particles bind to cells, if any of those fatty acids would actually induce or restrict binding of the minimal viruses to cells. So we compared something that we call the native spike protein on top of the viruses, essentially the recombinant protein as it comes out of the cells. And then this was compared in a second experiment with the apo spike protein, so essentially that protein where all the fatty acids have been removed and that was loaded onto the vesicles. And as we expected, actually in this configuration of the protein, we saw a higher binding of the minimal viruses to the cells, which is well in line with this up-down concept that I introduced you in the beginning, where the removal of fatty acids will essentially stabilize an open configuration of the minimal virion that leads to binding to the cell. If we incubated additionally a saturated fatty acid, palmitic acid, with the minimal virions, we could actually see that there was no essential difference between the apo spike form and those loaded with the palmitic acid. However, once we started loading unsaturated fatty acids, oleic acid, linoleic acid, and so on, we could see that the binding was significantly reduced back to the stages of the native spike protein. So from that very simple analysis that I just want to highlight wouldn't have been possible with natural viruses produced in cell cultures, we could already see that unsaturated fatty acids would reduce the binding of our artificial viral constructs to epithelial cells. And that was most likely caused by a closed spike configuration. Okay, that tells us about the function itself, but it doesn't really tell us about the reasoning behind why actually this fatty acid binding pocket is around. And if we want to understand that a little bit better, we actually need to look if this structure or this feature of the spike structure is a random thing, which is quite unlikely, or if it's something that is very well retained across different variants of the virus, and that we did by simple sequence comparison. You can see that here from the initial Wuhan strain and some other variants of concerns that emerged over the years in a sequence an alignment of the fatty acid binding pocket. You can see some of the crucial residues highlighted. And you might appreciate that none of them actually changed in the variants of concern that had emerged until then so far. So this kind of evolutional persistence and the conservation of the fatty acid binding pocket across different pathogenic strains of the coronavirus pointed us in a direction to say, this is an important structure. The virus seems to have, let's say, an interest in keeping this structure, and there seems to be an evolutional advantage associated to that structure, but we don't know really why. And to understand why this is around, it's actually important when looking at viral particles that are very much focused on reproducing and springing from host to host to consider the two most important processes in that sense. Yeah. The first important process that a virus needs to fulfill is being highly infectious. It wants to infect as many cells, as many hosts as possible. At the same time, enveloped viruses such as SARS-CoV-2 are basically know that they will be eliminated at some point. There's no interest, no evolutionary interest in killing the host. Yeah, it wants to essentially persist in the host long enough to produce new viral particles in order to infect new hosts. So it needs to have a high infectivity, but at the same time, a low immunogenicity. These are the two, I would say, or argue, most essential parts of virus biology. Well, now if we look at the immunogenicity side of the fatty acid binding pocket, we can first of all appreciate two things. If we look at the fatty acid concentration in the lungs of COVID-19 patients, we knew from the very beginning of the pandemic that fatty acid levels are elevated in the lungs of COVID-19 patients. And that is more or less unexpected finding because fatty acids are main contributors to inflammatory signaling. Essentially, polyunsaturated fatty acids actually induce inflammatory signaling in the lungs. So polyunsaturated fatty acids are a key signaling molecule in the immune system that would induce kind of an alert stage where the immune system is attracted, immune cells are attracted to the site of infection. So this led us to the hypothesis that the fatty acid binding pocket could actually, or the function of the fatty acid binding pocket could actually be linked to inflammatory stages. And it could be a mean of the virus particle to sense the local inflammatory state in the lungs of infected patients. So how to dig into this and how to use our minimal SARS-CoV-2 viruses to understand if this is actually a plausible hypothesis. So for this, we went again back to the structure of the spike protein that we solved, and we looked at the surface accessibility of the individual amino acid residues. So you can see here maps of the open and of the closed spike protein, closed on the left, open on the right side, and each amino acid residue is coded, color-coded in terms of its surface accessibility. So the deep magenta being very accessible, and essentially the white gray ones being not accessible. And for most of the spike protein, you don't see a change at all. But from the open configuration, from the side where the spike protein will bind to the AC2 receptor, we could actually see that there's quite some significant change in the accessibility of those residues, which makes perfect sense because they're more exposed in order to bind to the AC2 protein. So based on this, we looked at some very selected amino acid residues in this receptor binding motif of the spike protein. And the selected amino acids that we looked at were those that were known to be highly immunogenic. So essentially those that would most likely be bound by neutralizing antibodies. Those are also the amino acids that are frequently mutated in the variants of concern. And if we look at their surface accessibility in the open and in the closed state, we could see that the immunogenic amino acids are more exposed in the open state. Something that also makes sense in a picture where the virus would essentially try to hide its most important amino acids for the binding of the AC2 receptor by closing the spike protein. And once the virus needs these amino acids for binding of the receptor, it will go into an open state, expose these residues, and enable for the binding. So as the FAB controls the open and closed equilibrium in the spike protein, we could essentially say that the fatty acid binding pocket will control the exposure of these immunodominant sites in the spike protein. So that's a hypothesis that led us to test it if immunoglobulins such as IgG, in this case CR3022, which is one monoclonal antibody, which is also thought to be given Trump back then, actually, as a nice side note, essentially to test if this monoclonal antibody, which is known to neutralize the spike binding to target cells, if that neutralization potency would be influenced by fatty acids in the medium. For this, again, we applied our minimal virions, tested them in the individual fatty acid configuration, so with saturated and unsaturated fatty acids, and we could see that the neutralization potential of this antibody was actually decreased when the fatty acid binding, when the unsaturated fatty acids were incubated with the minimal virions. Similar effects we could also see when testing antibodies derived from COVID-19 patients, so from their serum. We could see that the infectivity of our minimal virions was reduced once we incubated these patient-derived immunoglobulins with them. But once we added the fatty acids, we'd actually see an increase back to initial levels almost. And now to close the circle and basically just tell you that these might all be in hypothesis, but to go back from an artificial assembled particle to a real virus, we also tested the influence of fatty acids, unsaturated fatty acids, on the infection of SARS-CoV-2 viruses in cell cultures by measuring the infectivity through messenger RNA levels inside the cells. And we could see that in a dose-dependent manner, polyunsaturated fatty acids will also reduce the infection of cell cultures treated with these polyunsaturated fatty acids. So this is a finding that could have been obtained with natural viruses easily. So everyone could have taken with a security laboratory, could have taken polyunsaturated fatty acids, incubate those with cells and viruses, and observe a reduced binding. But by the use of such artificial constructs, we're actually able to backtrack the mechanism by which fatty acids actually induce this reduced binding. So I just want to highlight that this was one example of fatty acids, of how minimal viruses, minimal virions could be used to dig deeper into biomedically relevant mechanisms for the infection process of viruses. So essentially a development that would include model 1 and 2 of that viral replication, but it's also the development that could kickstart essentially a technology that would also allow for the release of any kind of a cargo that is present in the artificial viral particles, and initiate the development of a technology that would allow for repetitive replication of such constructs inside cells. But admittedly, I think module 1 and 2 are also the easier ones to perform. And I just want to highlight how we envision to create such cycles of repetitive infection in artificial constructs. I think module 3 is quite easy to implement, since we have a lot of release modules, release technologies from the field of liposomal drug delivery. So there are pH-sensitive lipids that induce lysosomal escape, and that would induce the release of any kind of genetic information present in the vesicles. There are specific peptide, physiogenic peptide derived from viruses that would induce the release of any kind of genetic information. Also, module 4 seems to be quite easy to implement. We have decades of knowledge for genetic engineering and the artificial synthesis of genome structures that are able to induce transcription and translation, and integrating such viral genomes into liposomes is at least feasible, although challenging yet, but feasible in many applications. I will skip module 5 for a moment and just focus on module 6, which is the directed release of newly assembled viruses from the host cell. And that essentially would just include a module that prevents degradation of the newly assembled particles from the host cell machinery, as host cells tend to recycle every protein and particle that they find through a lysosomal pathway. And that's something that viruses, natural viruses, are very good to escape from. There are tons of techniques and methods to do this that could be adopted from the field of extracellular vesicle engineering. So, for example, there are technologies out there that can essentially reconfigure the escort complex, something very important for protein sorting in the Golgi and ER compartments of cells, something that could reconfigure the sorting mechanism in order to induce controlled and site-directed release of these viruses. So I think for model 3, 4, and 6, there are quite sophisticated techniques out there that could be adopted in this case. But I think the trick is actually, in the most challenging one, would be module 5. Yeah, that's the module that would close the circle, and that's something that's very hard to adopt. Yeah, because essentially the genetic information that you would need to put in the viral particle would need to encode modules that reprogram the biochemistry of the host cell, not only in a way to assemble new particles, but in a way that would allow to keep the quantitative control over the particles that are assembled. And once we go deeper into the biochemistry of a living cell, we also lose this quantitative control. So essentially, module 5 would need to be something that allows for the integration of artificial components. So, for instance, artificial lipids, artificial nucleic acids that are supplemented into the host cell cultures in a way that they could self-assemble into viral-like particles inside the cell that are then released back again through a controlled mechanism. That could be done through the artificial induction of phase-separated droplets, something that we propose in one of our perspective articles, where we know that some intrinsically disordered domains of proteins at least tend to form phase-separated droplets inside cells that can be used as production machineries for viruses. And it's something that natural viruses are thought to use in their natural infection mechanisms. But I think one of the other important things that I want to point out here is that module 5 is something that could, although it is challenging, that could be well benefit from bottom-up technologies, especially those technologies that focus on bottom-up assembly of artificial cells. So artificial cells could be used to benchmark technologies for the artificial production of virus particles inside a biochemically active intracellular environment that might be derived, for instance, from in vitro transcription translation mixtures that could induce this phase separation of droplets and so on. So I think module 5 is very challenging, and we still are out there looking for ideas how to implement them. It is the most challenging module, and this is the module that would close the circle eventually. I think another important aspect, something that we pointed out in the perspective article that we published end of last year, are the ethics associated with that. I introduced to you the motivation of ChatGPT given in the introduction. Essentially, if you ask ChatGPT if we should build these particles, there's no definitive answer about that. It's something that remains to be discussed, I think, especially the biosafety, biosecurity aspects. We give some considerations on these aspects in our perspective article. It's something that I think should be actively discussed by the community in future. Not only if we should assemble such or start to assemble such intrinsically infectious artificial particles. It's something that has been around also for quite a while for engineering or gain of function experiments for natural virus particles. But I think it's one of those examples where we as a synthetic biology community need to engage as early on as possible in order to guarantee biosafety, biosecurity for those artificial constructs that we built. So with this, I would like to thank the people that were involved in this project. That was mostly our collaboration partners at the University of Bristol, as well as the people at the Planck Institute for Medical Research, people at Heidelberg University and the Technical University of Munich, all the funding agencies, and you for your attention. If you're in Europe late at night, if you're in the US early in the morning, and I'm happy to take questions. Thank you very much.
Thank you. That was fascinating and really inspiring. There are several questions in chat. Can you see chat?
Yes.
If you could read the question out loud before
Sure.
answering, that would be great. So
first of all, someone is amazed that ChatGPT is starting to give talks now. I think we're not yet there, but it's certainly a tool that I can recommend everyone to play around with. I think it can help in the scientific process here and there. We shouldn't take it too serious, but just as a nice experiment, I'm happy to recommend that tool. So the first question is, could you please discuss a bit about the safety and security in my lab for this kind of gain of function experiment? So I'm trying to give a summary of what we proposed early on for the biosafety, biosecurity aspects. So as long as there's no genetic information involved in this that would give the particles an intrinsic replication capability, I think there's not much that we need to do apart from the standard safety procedures in terms of biosecurity, biosafety levels. We're working with recombinant proteins and so on. But apart from that, there's nothing that we need to consider specifically about this. However, once we start to engineer particles, so put together particles that could be potentially replicative, we need to consider this. And this is not only achieved once we start to put genetic information into the particles, but also at the point we introduce these systems and we interface them with natural cells, because natural cells can have viruses. There could be crosstalk between an artificial non-replicative agent, such as a minimal virion, and a natural virus. So essentially what we propose is a 3-level system for biosafety, biosecurity measures. That is, when individual modules are tested one by one, there's no special precaution. Once we put together 2 or 3 modules that would form a line in this, we would go to something such as S2 level, so something with higher precaution. And once genetic information and a potentially closed cycle loop is established, even though some modules might be missing, but that could be theoretically provided by the host cell, well, biosafety S3 measures would need to be considered. That's a simple summary of that. So the next question is, what type of viruses can you engineer? Respiratory only, or others too? I think that's a tricky question. I highlighted the role of membranes and our knowledge in the community about membranes, so especially enveloped viruses. I don't think that the site of infection is too important in this case. SARS-CoV-2 was an obvious example when we started this research. It's a respiratory virus, but there are other important viruses and lipid enveloped viruses around that are not respiratory. So I think the concept can be translated to other types of viruses as well. The next question is, would it be possible for a project that you use bacteriophages instead of human viruses and use it as some sort of antibiotic alternative? I think that's a very interesting question. I'm not an expert in bacteriophages. As far as I understand, they heavily rely, as many other viruses, on self-assembly. So many bacteriophages can actually be self-assembled in vitro by the simple protein-based building blocks that form the capsid. And I think it's something that could be implemented as well. Their use as an antibiotic alternative are discussed heavily in the last 1 or 2 decades now, at least in the Western world. But I think it's something that this line of research of assembling artificial viruses should not be blind to. Yeah, it's the example I'm giving now is for those of enveloped viruses, but there are many viruses around, not focused on mammalian cells, but also infectious for prokaryotic cells that we could think about reconstructing. And the advantages would be the same as for the enveloped viruses, right? We have a high degree of control over the structure and the function of these viruses. So the last question I can see in the chat is, virus functional annotation is not as evolved as cellular organisms. So what sources do you envision using as the genetic pool for a minimal virus? So if I understand the question correctly, it's mostly about how we design the genome of such an artificial virus. And that's something that, I think I got the question right. And so how do we design the genome of such an artificial virus? So the engineering of viral genomes has been discussed as well a lot in the past. There are some approaches. There are very limited approaches on gain of function design because there are also ethical considerations associated with that. But I think in principle, the design of a minimal virus genome should follow this modular concept, right? So there should be elements that would engage the in vitro transcription translation machinery. I think for the initial part of that, we should not be focused too much on compressing the genetic information. That's something that viruses are especially good in, putting a lot of function in a very limited amount in a very short sequence. I think as we use artificial approaches, we can be a little bit more flexible with that and go for larger genome sequences that are more flexible and more adaptive. Those viral genomes should also have intrinsic structural elements that will allow for a packing into the newly formed viral capsule, something that natural viral genomes are also especially good at. There is some structural proteins around that would essentially compress, spatially compress this information again, and there are very specific sequences that are recognized by these proteins. So I think that's a specific model that should be present in there. And obviously, that's probably the most important part. There should be a coding sequence around that would allow for a new module that combines the function of module 1, 2, and 3. So something like a spike protein encoded in there. I think that's to start off with kind of a very primitive design, but something that could be enough to kickstart such a viral replication cycle in human cell cultures. Okay, that's everything I see.
That sounds great. I thought that was already answered. It was related to the safety security precautions, and that's I'm really glad to hear that explanation. If there are no more questions, I want to say thank you so much, Oskar, again.
Thank you.
I'm really interested to see how that work develops. Thanks. Thank you. And
thank you very much.
Great. Thanks, everyone. And see you all next week.
See you. Bye-bye.
Have a great week. Bye.